Today we are here at the Thoraxklinik Heidelberg, one of Europe's largest centers for lung health. The clinic plays an important role today in advancing the understanding and treatment of Alpha-1 antitrypsin deficiency, making it the ideal place to revisit its fascinating history. And who could better guide us through this sad history than these two distinguished voices? One we have here from Hannover, Professor Dr. Sabina Janciauskiene, who is a well-known pulmonary researcher with over 2 decades of expertise in the acute phase protein Alpha-1 antitrypsin, and Marion Wilkens, a dedicated patient advocate and leader of the Alpha1 Germany patient organization. Together, they will guide us through the science, the people behind the discovery of Alpha-1, and the remarkable story of how observation, technology, and clinical need sometimes not only depend on smart minds, but also on the right timing. After a short walk through the Thoraxklinik Park, we are here at the library of the Tuberculose Museum. A room full of history and the perfect place to answer a few questions about the history of Alpha-1 with Marion and Sabina.
And Sabina, it is a pleasure for us to have you here, and thanks for doing the way all the way down from Hanover to Heidelberg, and to let us know about your knowledge and your experience about the history of Alpha-1. And I think it's a lot you know about that, and thanks for sharing that.
Yes, thanks a lot, Marion. Thank you for the invitation. And it's a great pleasure for me to be together with you and share some ideas about Alpha-1.
Yeah, and before we start with the history, we should even let the people know what is Alpha-1 and what does it do in the body.
Yes, and Marion, thank you for question. This question we always ask each other, and you know my answer. It's my favorite, most beautiful protein, multifunctional protein, and a lot of scientists have a large interest in this protein until now, right? And if you're seriously telling, this is one of the most abundant proteins in our body. It's after albumin, immunoglobulins, it's the largest. It has about 1 and 2 grams per liter of blood, so it's a lot. And it is also a very important protector of our lungs from the proteolysis because it's a major inhibitor of neutrophil proteases, like neutrophil elastase, but also has multiple other functions, right? Immunomodulator, anti-inflammatory, and it is acute phase protein. Therefore, it's protecting us during infections, during inflammation, when the concentration can rise above normal, even 3-4 times. And then we have a protection level, right, of antitrypsin. We know that about 0.5 gram it is per liter would be still protecting us. So I would say this is short about antitrypsin, right?
Sabina, when did scientists first discover the protein?
Yeah, this is according our written knowledge, if it's really true. The whole story begins by the end of 19th century, because not until this time we knew that in our blood we have proteases and protease inhibitors, because there are no methods available to isolate proteins. So the first written message comes from around 1863-64 from the French physiologist Astre, who described that in our blood we have proteases, right? And then the later Italian scientists, almost a similar years, they also notified, there was Fermi and Pernozzi, that in our blood we have also inhibitors or modulators of protease activity. So then it's, so to say, the first knowledge we have that we learn to know what is inhibitor and protease, but we didn't know how to isolate them. And then only in the 20th century, in 1930s, scientists become interested to develop techniques how to isolate proteins. And a large contribution comes from the Uppsala University from Sweden, when Arne Tiselius, he started to develop with other colleagues this specific electrophoretic technique, and for which he got Nobel Prize in 1948. And this technique was allowing us now to separate different proteins from the blood. And this was like a first, so to say, step to learn about antitrypsin. And then it was a kind of this technical capacity. It was not enough to discover antitrypsin, right? But it was a coincidence that in Malmö, in Sweden, Bertil Laurell, he was a biochemist and a very great scientist, and he developed clinical chemistry laboratory, and he integrated this and simplified even this complex technique for isolation of proteins, and added or employed this into the clinic, and started to analyze number of samples from the tuberculosis clinics, in fact, also. And then he discovered by accident, because you can, in electrophoretic field, proteins migrate dependent on the charges, and then they see each band of the protein. And then suddenly they see that Alpha-1 globulin protein band is missing. So as described Bertil Laurell, he thought that this is maybe, as happens sometimes, you get damaged plasma, or he thought it's mistake. So he requested to repeat sample. So the next day, doctors gave him samples again, and again he's running the same 2 patient samples as he described himself, and he again cannot see these bands. And then he thought originally, it's very interesting, that he thought it's another protein, because in the same fraction you have orosomucoid, which is also our blood protein. And he checked for that protein, but it seems to be okay. Yeah, it is still present. So then it was also very interesting, because I jumped a little in front, because similar time, 1950s, it was also when the electrophoretic technique came, when we understand that we have different proteins, scientists started to isolate these proteins, right? Develop different techniques to get purified proteins. And then 1955, already in 56, there were a first isolation of antitrypsin, right? And why we call antitrypsin? Because when protein isolated this, they see that it inhibits trypsin enzyme, and therefore it got the name antitrypsin. And then, so when Bertil Laurell excluded orosomucoid, he also got the same time, it was thesis published, and also these publications came that, yes, it is Alpha-1 antitrypsin should be in this position. So here we got the first observation that we have Alpha-1 antitrypsin, and there are patients which are missing this band.
After they discovered the missing band of Alpha-1, how did they connect that to emphysema?
Yeah, because first of all, in those first 2 patients, when they saw this missing band, they were coming from the tuberculosis clinic, right? And they had a lung disease, they had emphysema. And then they were, of course, rose a very large interest what is about. So then Bertil Laurell at that time also had his young physician student, Sten Eriksson, who was very interested in this question and subject. So they also looked for the family members, for the other patients, right? And then very quickly after those two patients were discovered all. So it was one family which had this deficiency of the protein. And then, of course, it was logic to assume that some people, especially with lung disease, with lung emphysema, they have a deficiency, seems inherited deficiency. And all this first summary of these first patients were summarized in the thesis book by Sten Eriksson, right? And that's why we say that antitrypsin deficiency discovery and connection to emphysema came in 1963, together with the publications and with the Sten Eriksson's all described book, showing missing bands. So that's how it came. And also maybe important to mention that it was a kind of right time for this discovery, because in 1960s or 58, this time was also definition for emphysema accepted, right? It was the first time that emphysema was included in chronic obstructive pulmonary disease like a subtype, like subclinic. So therefore, we said the inherited deficiency is related to emphysema. That is how it came. And maybe I will mention very short here that at very close, just a year after this discovery, because antitrypsin is inhibitor of trypsin, so scientists wanted to experimentally prove that missing of this alpha-1 antitrypsin would create problem to the lung, could create problems. So then they were using papain models, different enzymes in rat models, and induce lung damage. And then they saw that the enzyme, if it's too much enzyme, it will increase damage. So they also started at that already time to build this hypothesis that missing inhibitor will allow protease act more aggressively and induce lung damage. So that's how it was connected with this very nice book, right, written by Sten.
And till then, the physicians thought that it might be only for smokers to have an emphysema, and nowadays we know we can have an emphysema without smoking.
Sure, that was right. Nobody till then was thinking that it could be emphysema related to our own missing protein, even if the person is not smoker, right? So yes, this was very large breakthrough. And if historically we see this, it was not so long ago, because all the major discoveries, even elastis was described first in 1967, right? It's pretty short historically for us. And then also this, with the Lung Association came in 1963, before that.
Maybe one question, how important do you think it was that the definition of emphysema and the discovery of Alpha-1 took place at about the same time?
I'm now thinking, because to build the story, right, it's all events usually follow each other, because once you open something, then you can develop. So the opening was electrophoresis, opening was introduction of this method to the clinic, then finding some proteins and first you find the protein, then you find that you can have a deficiency of this protein, and then at the same time it's in the clinic development. So you have a definition for emphysema specifically. And here it comes a connection, right? That emphysema is already questioned. Until it was realized more broadly, it took years, even until now, right? But at that moment, it was that you have emphysema, which is not only smoker's disease, but also could be caused or could be promoted by genetic factors. Yeah.
At first, Alpha-1 antitrypsin deficiency was mostly seen as a lung disease. When did that change, and when did the liver come into play in the discovery?
Yeah, thanks. It's a really interesting question because I know from Sten Eriksson, because he was my supervisor, right? He was telling me that already when they discovered that antitrypsin deficiency relates to the lung disease, they also thought about liver, because it was known that antitrypsin produced by the liver, basically. And they thought it was a large discussions between in the clinic between the physicians and scientists, and they thought maybe it's because liver have some damage and is not producing this protein. So, but of course, the interest in Alpha-1 was not only in Malmö, but in all other universities worldwide. And then during these all discussions in Sweden already, Professor Sharp, or Dr. Sharp in New York, in the United States, he proven using from the children, newborns, that there are some newborn children which are developing jaundice, and they have cholestatic liver disease, and some of them even have liver cirrhosis. And then he connected this to Alpha-1 deficiency because he saw that these children have intracellular in the liver inclusion bodies. So then it came that there is some defect, that protein is produced, but it's not secreted in the circulation. And this protein intracellularly accumulating in the liver can cause this liver disease or damage in the liver. And he published this work first time in 1969.
Oh, now it makes sense, because I was born in 1969 and I had a severe jaundice, and nobody knows why I had this, and nobody could help me at that time. But now it makes it clear why nobody knows it, because it was all only discovered in 1969 then.
Yeah, right. It was just published, and it was amongst some very small group of scientists was discussed that inherited antitrypsin deficiency can cause this liver damage. But in the broad clinics, the knowledge usually comes a bit later, right? You need to spread the knowledge. So it is very right. You're right. It was, as physicians didn't know, right? And children physicians, when they were newborn with jaundice or some cholestatic problem, they were not connecting in most of the times to antitrypsin deficiency at that time, right?
Hopefully they know it now.
Yes.
After they connected Alpha-1 antitrypsin to lung diseases and liver diseases in children, how did they proceed? Like, how did they find the data for further studies on the mutation?
Yes, it was a large interest in the scientific community about antitrypsin once these discoveries came from both lung physicians and also from the liver. And here in Sweden, also coming from Bertil Laurell's original findings and Sten Eriksson's, there was an idea to make a large population-based screening. And here was involved another young, very fantastic person, Thomas Sveger. He was pediatrician, and he took responsibility to carry this study. So study began in 1972 until 1974. During this time, here in all Sweden were screened 200,000 newborns. And in this cohort study, it was discovered more than 100 disease, and some less number SC, or let's say homozygous or severe deficiency and less severe deficient cases. And was also, this study gave us pioneering knowledge about the prevalence. Then it was calculated that in the Swedish population, you have severe antitrypsin deficiency. This we call ZZ deficiency, which is 1 in 2,000. Later was confirmed that in Europe, in different countries, are 1 to 2,000, 1 to 3,000, the prevalence. And also this study was, even until now, it's very unique study. It's a longitudinal study, first time giving systematic data on the natural history of Alpha-1 antitrypsin. And also it's raised a lot of questions from both ethic. Should we really do screening, right? Ethical question is debated until now. And also it gave suggestions or in the clinics how to take care, because this longitudinal study also shows if the persons grow up in the better environment, they are not smoking, they have exercise, they change maybe lifestyle, you will be a long life healthy. Most of or many of those cases were healthy. So it also gave us the first some knowledge for potential prevention medicine for this group of individuals.
This is what we always say. If you have the knowledge about the disease, you can do something for yourself. So this is what the Swedish cohort could do, because they know early on that they have the disease.
Sure, because majority of people knowing they will not choose to be cigarette smokers. They will maybe not work in some jobs where you have high pollution, right? They will choose the living conditions where you have more clean air. And the result is very, very good.
Mm-hmm. So Sabina, you talked earlier about PIZZ and PISZ, which are variants of the mutation. Can you explain this category system or variant system and when it was first discovered?
Yeah, right. This is a good question because I already mentioned Z, but yes, in the first, again, this goes back to clinical chemistry, to Bertil Laurel, because he was the first who introduced this name PI, which we often see everywhere. It means a proteinase inhibitor, because Alpha-1 antitrypsin is inhibitor of proteases. So it's called PI. And now, which variants of this PI? And then again, it was used specific electrophoretic technique where the protein migrate like antitrypsin. And if it has mutation or some change, the protein will get nullified, the charge, because protein migrates according to the charge. And if the charge plus and minus is 0, then the protein stops. And then different mutations, they will change this charge. Then proteins stop in the field at different positions. And therefore it was named normal, M, which is migrate normally. So it's a stop and right position. S is slow. It has mutation, which is stops at shorter distance. Then you have F, faster. And you have zeta. Zeta is very slow, very, very slow, specific. So all alphabetic letters was mirroring the position or migration speed in the electric field of the proteins. This is an old system, and it was originally first published in 1967 with Bertil Laurel and a student Fareholm from Norway. This is the first publication. So since then, we knew the variants, original variants. Today we know many more.
So now it is clear that it's genetic, and we also know the different variants of the mutation. What were the next steps for the scientists after these discoveries?
Yeah, it is true. The next, knowing that we have different variants of antitrypsin, it of course comes genetic interest. What is behind? And here was also very nice observations. One from Sweden, which was characterized Z mutation. Jeppsson professor, who described also found the specific one amino acid mutation in the antitrypsin, which causes this slow variant called Z. And then at the same time, another very common clinically known mutation, S, also slow, was the same year discovered or described by Robin Carroll in Cambridge. So the recognition of the future characterization of variants now came not only by migration in electrophoretic field, but also knowing that behind we have a single amino acid exchange, and therefore protein is changing. And here, of course, this also raised many questions about the history. I mean, why we have these mutations, and a lot of discussions. And I personally myself, here we have a book of Ignacio Blanco, and he also writes a history, biology, and pathophysiology of antitrypsin. And we talked a lot, and Ignacio even created some small video and shared with me about how origin of the deficiency variants comes. So simply to tell, this is from our current genetic DNA haplotype investigations, it is clear that Z mutation seems to arise from the Scandinavian part, and it could be about 120 generations back, about 2,000 years back, from the time of the Vikings, where the Scandinavian, Danish, Norwegian, Swedish, they had developed boats at that time, and they were travelers, and they settled, and they traded, they stay in different places. So this Z mutation, we can see gradient from the north of Europe, goes to Central Europe, to Germany. They also travel to Russia. We see in France, it reaches Iberian Peninsula, and also England, North America, this whole region. But we have even older mutation, which is S, which originates more from the south, from the Portuguese, from the Spain, and it's accounted now for almost 800,000 or 14,000 years back. And we also see gradient. This mutation is much more common in the southern part of Europe, Spanish, Portuguese, and then gradually a bit lower when we go to the north. So this is our kind of the origins, what we can account today.
Mm-hmm. Sabina, you just told about this famous book of Blanco, and you have shown us as well the first book ever about Alpha-1. Are there more books, or is there more literature to rare disease, or is it also rare?
No, it is not very rare nowadays, because there are several books, and specifically book chapters by European Respiratory Society. And we have our in German group wrote several chapters. There are also books. There are a lot of publications, scientific, from the pure clinical pathophysiology, from different aspects. So as science is developing further, this was the beginner, and now this goes. Of course, one always, when the new discoveries are coming and new observations, one would like to see good summary of that. Everybody would see it, would spread the knowledge. That knowledge will not stay isolated.
So whatever we are not understanding now, we can look it up.
Sure.
Oh, fine. Thanks.
To kind of bring back the timeline of the history, what happened between 1966 and 1989?
Yeah, this is a period of time when protease, anti-protease imbalance theory was built up. Because this theory was kind of very important to explain why some individuals carrying severe inherited deficiency develop lung emphysema at an early age. Knowing that antitrypsin is the fastest and the most strong inhibitor of neutrophil elastase, and knowing that neutrophils play a very important role during lung inflammation, acute lung inflammation. So during these years, a lot of scientists across all the world, from Europe, everywhere, in the United States, were very much focused, both clinical and experimental scientists, on this aspect, this anti-proteinase imbalance. You're missing antitrypsin, you have more activity of uncontrolled enzymes, and therefore the theory was that these enzymes start to break lung tissues and damage alveolus, and you get emphysema, empty lung, so to say. So here I cannot mention because there are many great scientists involved, and difficult to exclude or include one, but really were a lot of experimental models created in mice, in rats, and other, also in the vitro experiments and the clinical studies, of course, to prove this theory. And this theory actually gave the idea that we can supplement this missing protein in a kind of correct balance biochemically. Right? Maybe if we isolate protein from the healthy individuals and give the one who is missing in the circulation, then we would balance this kind of aggressive, we balance elastase or other proteinase activities. So, and here the contributions came the first from United States, from Ronald Crystal group, where they were isolated. They were actually, first publication came in 1989. It's by the end of this whole period of where they isolated more than 300 donors antitrypsin, and then they used it for the patients as a first clinical trial to supplement this protein. And at the same time, they also got permit from FDI, from American Drug Administration, to implement this treatment to the clinic for the patients who have severe inherited deficiency, and they have developing emphysema. It was even created guidelines, which we know today, and today they improved these guidelines, when to treat, which way this patient should be absolutely not smoker, and so on. So then this 1989 period of time ending with the introduction of the drug. And then this time was very important role for the biotech companies. So Kate Biologics was the first, which is now kind of integrated during the years. It's part of Grifols, our largest producer of plasma products and also provider of Alpha-1 antitrypsin for augmentation therapy for those patients who need.
Yeah, and Unfortunately, the augmentation therapy, so easy it sounds, is not available all over Europe for everybody. So we have lots of countries where it is not reimbursed. And this is a shame, to be honest. It's not understandable, or?
Yeah, you are right, Marion, because it is so far only one specific therapy for this group of patients, because it is specifically augmenting missing protein. Until now. Now it's a new therapies may be emerging, but this is specific. So logic says that if in one country available, so it should be in all European countries available for those patients who really need this or they want to choose to have this therapy. You're right. We hope that this could change in the future, that the therapies, specific therapies could be available for those who need it.
And if you want to know more about the history of the therapy, I think it's a good place to go to Barcelona, to the Grifols Museum, and it shows very clear and very good the history, how it all began in that case.
Oh, sure. Very good suggestion.
We go there together.
Yes.
Sabina, in your first sentence, you said that Alpha-1 antitrypsin is very special. Is it really as specific, or are there other proteins as well?
Yeah, it's special for me. But in general, you are right. Antitrypsin belongs to the family, which is named as serpins, serine proteinase inhibitor family. And this is name given by late 80s by scientists, including Robin Carroll and many others in the world, where it was discovered by looking at the protein structure and function that there are a group of proteins which share, some of them maybe 70% of homology in the structure, and they all are inhibitors of serine proteinases, like, for example, antithrombin, everybody knows, or antiplasmin, or some. So all these proteins were connected to the serine proteinase inhibitor family, with antitrypsin named as an archetype member of serpin family. And then it also became acknowledged that other members of this family, for example, neuroserpin, they also can have point mutations, inherited mutations, which also can cause disease. For example, neuroserpin mutation can cause brain-related diseases. So therefore, there was another name introduced by Cambridge people. David Lomas, Robin Carroll suggested to call this serpin-related disease serpinopathies. So we have one more word in the literature, serpin, and serpin-related disease, serpinopathy.
So now we know we have different mutations, but what is happening with these different mutations regarding the protein?
Yes, it is this is the next large area of investigations, which also started in the 90s, right? And it's continuously ongoing, because what we learned to know that this deficiency of some mutations like zeta mutation causing severe deficiency, it is not because the protein is not produced, but it's because the mutation changes folding of the protein, changing structure of the protein. So protein start to build aggregates, which is each other. It builds so-called polymers. And then they could be very long, very short. And I must acknowledge that this polymer structure for years, decades, strongly studied Cambridge group, Robin Carroll and David Lomas. They are really experts in explaining these polymerization mechanisms. And therefore, if we have those two interconnected diseases, because liver produce mutated protein, and protein is not correctly folded, and it builds polymers, and it accumulates in the liver. So we call it gain on function disease, right? Because you have protein stuck in the liver cells. It could cause a problem. On the other hand, protein is not secreted, and secreted also polymerizes. It also builds these polymers because it's inherited property of these proteins to build these polymers. So then we have loss of function. So we have lower levels, but even those levels, because they are not natural, but they are polymers, they also have a reduced functionality. There are a lot of studies analyzing how these polymers could also contribute to the lung disease and how they could be pro-inflammatory and so on. So there are a lot of, in this time, so to say, the second time of understanding of antitrypsin-related deficiency diseases, it comes this structure, protein structure biology and structure-function relationship. And I would like to be proud a little bit because when I defended my thesis, I was focusing on another form of antitrypsin, so-called the fragments of the protein, because antitrypsin is inhibitor of elastase or other enzymes. And when enzyme interacts with inhibitor like antitrypsin inhibits protease, but at the same time, it's irreversibly damaged, it's cleaved. And then the small fragments of antitrypsin are generated. And originally we thought that these fragments are not present in the circulation. But today we are sure can find them and show them and relate to specific diseases. And in my thesis in 1996, the same, like in 1996, Robin Carroll and David Lomas published this critical work about polymers. I was also pushing the theory on the availability of the fragments and their importance for the patients of the fragments. And also would like to mention the point which is important for me personally, and I thought it's interesting to know that during my thesis, there was a large presentation, and my supervisor was Sten Eriksson, who discovered antitrypsin. My opponent was Robin Carroll, the polymer person. And in the audience was sitting Bertil Laurel. It was very rare. I knew from other colleagues he will come to listen for thesis. And I was very proud. It was in my scientific career or scientific road. It was one of the most beautiful moments where different generations involved in antitrypsin studies were together, where the original discoverers were opponents, and where I myself was my new, so to say, suggested theory. So this is a beautiful moment, and I wish that this moment will happen for other new coming students, for people where you can be together with elderly who made certain observations, and you continue work and you contribute even better to, even with novelty. So this shows this transition of the generation, and this kind of encourages me to promote collaborations very much.
So you are really living history.
Exactly. And it also shows how young the history of Alpha-1 is, because you could all come together in one room.
Right.
The start of the history and with your thesis, the evolution of the research.
Yes, that exactly shows the young research, but also have a lot of achievements.
Exactly. You want to show your thesis too? That's your thesis, right?
Yes.
Yeah.
Yes, this is a thesis. Hydrophobic interactions of serpents. And among them, it comes fibrillar-forming hydrophobic peptides of antitrypsin.
So there was a lot of research and a lot of progress made in a very short time. how does it help patients today, and why is early diagnosis so important?
Yes, this is the most important question comes because history per se would have no value. It definitely, for patients, since we know now this deficiency condition, so and it's treatable condition. Therefore, early diagnosis, it is very important. And therefore, the screening also important. And but here we often say that nowadays antitrypsin deficiency is rare. It's not a rare disease, but really rare observed or diagnosed. And because the condition is could be treatable on one hand, but on the other hand, many individuals carrying this mutation would remain long years, large part of the life, and asymptomatic. They will not show symptoms of liver and lung disease, maybe later in life. But if we also know, as I mentioned earlier in our discussion from Thomas Weger's study, if you know that you have mutation, that you are healthy, but you care, so you prevent disease development yourself and with the help of physician. So therefore, nowadays, organizations like World Health Organization, European Respiratory Society, American Thoracic Societies, and there are very clear guidelines and recommendations that antitrypsin, also PD patients or emphysema patients, they should be screened for antitrypsin deficiency, especially if the person developed disease at younger age. But unfortunately, from my knowledge, not every clinic or every physician still care about, because the one reason is that the symptoms for Alpha-1 patient, if he is really starting to develop disease, for example, first bronchiectasis, asthma, or first signs of chronic obstructive lung disease, they will be not specific for Alpha-1. They will be typical, like for all patients who are not carrying this mutation. So therefore, this one also reason when one could be misdiagnosed or simply not even cared about this mutation, that you should check for this mutation.
Yeah.
So this is what we need to always educate physicians and to look at mutations.
And it's not only on the lung side, it's also on the liver side. Often it is said to us, please stop drinking, please stop eating fatty things because you have a fatty liver. And nobody cares that fatty liver symptoms are the same or nearly the same as we have when we have Alpha-1 disease. So yes, that's a very big issue for us, that there are symptoms which many patients have, and we are just a small group of them. And it is so easy to think, just do the test, do the serum level, and you know more. And this is what we always have to carry out in the world.
Exactly.
Yes, so therefore, according to the current knowledge, we could say around the world, we have about 10% of deficient patients only recognized or known. The majority of patients are not patients, carriers even, individuals are not recognized or not tested for.
And it takes up to 5 to 10 years till we are really diagnosed with the right diagnosis.
Yes.
So that's really still a big problem, even if we know so much from the history.
Yeah, sure, because a lot of patients, from my interactions, also telling they need to pass several doctors, sometimes 5 or more, until the right diagnosis comes, until you learn that behind you have inherited deficiency of important protein, and for lung or for the liver.
So awareness is one very important thing to have the disease known by the people affected maybe, or the condition as you described it in a more fitting way. Marion, as the leader of Alpha1 Germany patient organization, what role do these patient organizations play in bringing this awareness, and how did they historically bring that awareness?
Yeah, this under-recognition is really a problem where we are coming into the play, because it's good to have patients who have a strong voice and to show that we still need more research, more knowledge. And this makes our works very vital. So the first, coming to the history, the first patient organization was 1995, I guess. It was the foundation in America, the Alpha1 Foundation, founded by John Walsh, and I'm very proud to have met him. I think you have met him as well. Yes. He was a very strong person, and he had done very lasting awareness and knowledge and research. He has done a lot for the Alpha1 community. So he was very a starter for us, and we all look to him and say, lovely person, good start for patient organization. And years later, many organizations all over the world comes and built or was founded, and like Alpha1 Germany, founded in 2001. So it's also a long time ago. And since then, we really try to bring our knowledge to the world, or the knowledge of the physicians to the world. So we want to help the patients and to bring back their better daily life, and we help as well the relevance and the family to have a better life. As one of our goals is education of patients, education of physicians, and education of the general public. We need really a good network to do this. And we need all these people in the Alpha1 world which helps us. Like we have a good advisory board where you are in, Sabina. And we have also a good connection to other organizations. I would like to mention here some examples, like we have good connections to the DGP, to the European Alliance, and to the AXE, as well as to DZL. So we have really good connections all over the world, and especially also in Germany to other patient-related or lung-related and liver-related organizations. And this is very vital for us, because we can only do together something for us patients.
Yes, I would also like to add one momentum that, according to my knowledge, German patient organization is one of the largest, and many other organizations very much, one can say, even dependent of follow-up development of German patient organization.
Yeah. With the rare disease, we have more than 1,100 members. So this number is telling something. Yeah.
Yes. And this is really like a guidance, guiding association for others. So,
and we also want to be Wikipedia for Alpha1. So this is, you always can look on the website. I think we have mentioned every point of which is necessary to know about the disease. And this is also a good goal for us to be Wikipedia.
Yes, fantastic. Yeah. And this is also shows how important are collaborations. Yeah, how important is network connections with everybody, and it takes time, energy, and it is good team. So having good team, it is a power.
Yeah.
I think the engine of our success is really the good relationship we have to our physicians, to our researchers, as well to the patients. So the combination of all these stakeholders,
The connection, yeah.
the connection, this is what is the success for us.
Yeah. It's really interesting that or really nice that you mentioned that with the also ongoing research. Maybe Sabina, you can tell us something about research that is now been done by scientists.
This is true. Ongoing research very fast and in multiple directions nowadays in antitrypsin, really, because it's, as I said, it's attractive protein for research. So first of all, I already mentioned, we talked about we have augmentation therapy for deficiency patients. And this therapy is, for the moment, is a plasma-derived product used intravenously. But nowadays, it's also developing this product. There are other applications. Scientists are trying to develop inhalation therapy using antitrypsin, subcutaneous therapy. It's also idea is not to use always from plasma purified protein, but also produce recombinant, which can be maybe easier to produce and will be no risk for contaminations from plasma derived. And so this is even augmentation therapy, which is already long history, it's still under development. But we have also new therapies are coming. For example, we talked about that antitrypsin have this conformational or structural change, building polymers. So there are also developments of small molecules which could prevent this aggregation of the protein, which it would allow to release from the livers or in the plasma. We have also, if we say we have deficiency of the protein, then we assume we have more proteolysis, as we talked, elastase activity. So there are also developments in the small molecules to inhibit elastase. So instead of antitrypsin, we can produce synthetic molecules nowadays, which also will mimic, so to say, antitrypsin effect and inhibit proteases. And they are more sophisticated because of the current developments in the genetics, in the molecular biology methods. So there are new therapies coming which I would aim at the gene correction, even to correct the gene to produce the right protein. Or even when the protein is already transcribed or expressed, correct this with interference RNA or with the silencing. You can silence protein, so the gene remains, but the incorrect protein is stopped, so it's not produced. And here again, then it comes importance maybe for the combination. Then you stop not good protein or defective, but instead you can supplement or augment individual with a correct protein. So there are many new ideas and new approaches coming for the future.
Yeah. And what would you say, why is it so important that this research also does not stop at one point?
Yeah, this is a very important question. Summarize what we discussed, because we can't stop. Because our aim is that people who carry this mutation or have this condition, that they would not get the severe disease. This is our aim. And they could be diagnosed early, that they could be treated efficiently. Simply that we will say we have a mutation, but we can care, and no worry, you will always be healthy. So therefore, we cannot stop this.
No, and we take hope from these researchers. So this gives us hope that in future, maybe we find a cure.
Yes, it's with the modern gene therapies, with the modern approaches, technological developments, we have a large hope. And even then, why should we stop? Because the protein is a member of the large serpin family. And this protein will also give guidance to approach other diseases, other disease areas, and maybe antitrypsin as a biological, also therapeutic, could be very valuable, not only for Alpha-1 condition, but in generally. So I guess we cannot stop.
No, never.
And the reason why we should not stop our research is one, also very important, because we continuously recognizing new variants, new mutations of Alpha-1. And currently we have more than 500. And also only part of those, maybe less than 100, where we could say today they have some clinical relevance. Many we cannot connect to the clinic. So I think this is also gives us very strong reason to continue research in Alpha-1, to continue search for new mutations, and also to understand what these mutations means, what role it has. Can we use this knowledge for future development?
So Sabina, we started in the late 1800s from the observation of the protein to the therapy and ongoing research. I think it's still a young history, but very full of a lot of knowledge and a lot of important work from a lot of scientists included. What would you say, what would be your message to future researchers or to patients in the Alpha-1 community and health organizations maybe in general?
Yes, I think what I could tell shortly, that we got the gift from our history, from starting from Alpha-1 antitrypsin as a protein discovery, to understanding that we can have inherited deficiency, and then to therapies and very advanced therapies nowadays. So I wish to all of us and patient organizations carry on this gift and develop future research, organizations, collaborations, all together, because we always have expression, student should be better than the teacher. If so, then the field will continue to develop. That I would like to wish.
Very nicely said. Yeah.
Thank you, Sabina, for being here. Thanks for sharing all your experiences and your history. And we both will never stop.
No, never.
Never.
We should continue and continue.